Good afternoon, biology. Today we're going to be talking about energy and enzymes. So, energy just means the ability to do work or the capacity to do work. So, we have different types of energy. We have kinetic energy, which we say is the energy of motion, and then we have potential energy, which is the energy of position or the energy of composition. If we're talking about chemicals, it's more about composition. If we're talking about objects and gravity, it's more about the actual position where it is. So if we look at these two people, one of them is holding a ball, the other one is dropping the ball. So her holding this ball in position, it has energy based on where it is. So it has potential energy. Once it's actually dropped, well, then it's energy of motion, so it becomes kinetic energy. Thermodynamics is the study of energy. Specifically, we think thermo, we think heat. It's the study of heat. We say dynamic, we think of movement. So it's these energy transformations, these energy movements in the form of heat. This unit we're going to use is the kilocalorie. And a calorie, the definition of that is the energy required to raise 1 gram of water 1 degree Celsius. When we talk about food calories, a food calorie is actually 1,000 of these calories. So a kilocalorie is 1,000 calories, and 1 kilocalorie is 1 food calorie. And I put this little image here. It was supposed to be an animation, but for some reason it's not working. You can see the heat coming off of people. Now, why is that happening? They are burning molecules. They're breaking down molecules in their body, and that's releasing heat. It's their metabolism. And that's why you can see that energy coming off of people. They're literally burning calories. So if we think about how we measure these calories, I just looked this up just out of interest. Love McDonald's, but their food is quite calorie packed. 1 triple cheeseburger meal at McDonald's is 2,000 calories. And this is the kind of cheeseburger sandwich that's not even like their premium quarter pounder patties. This is like the smaller ones. So I can't even imagine what that one would be. It must be close to like 3,000 calories, I would think. So that's a lot of calories. And these are food calories, capital C. So you can multiply that by 1,000 to get your actual regular lowercase c calories. So we talk about chemical reactions. That's going to be important in terms of heat transfer and how heat will move through a system. So again, a reaction is when you make molecules run into each other. They literally run into each other and collide, and the atoms are rearranged to get new molecules that did not exist before the reaction took place. That's how we know we have a chemical reaction, if we got new molecules from the mixture of those molecules and the reactants. So we call what we mix together, these are the reactants, and then what we get on the other side would be the products. And in order for it to be a chemical reaction, you have to have new molecules formed, right? CO2, H2O, we're not part of the reactants. And usually what's going to happen when that occurs is you're going to have a color change. You might have a temperature change. Now, the energy to make this happen, we call activation energy. The energy that we have to add to make the reaction start. So one of the types of chemical reactions is if heat is released, we call that an exothermic reaction. So reactions that release heat, we say, are exothermic. They call them spontaneous because they generally occur without, like they occur on their own more easily, so are spontaneously. Sometimes you'll hear it called thermodynamically favored if you've taken AP chemistry. They might call them catabolic. So what's going to happen with these is the reactants will have more energy than the products. And the reason you have that heat coming off is because the products don't have as much energy, and all that energy can be released as heat. So endergonic, the opposite of exergonic, is endergonic. You might hear it also called endothermic. These absorb energy, so they're going to feel cold. The products will have more energy than the reactants. And that's why they feel cold, because they're absorbing heat from the surroundings, so they feel cold to the touch. So an exergonic reaction is one that is going to have less energy in the products than it did in the reactants. So that difference in energy is released as heat. If it has an enzyme, enzymes lower. You see this bump right here? That's called activation energy. If you have an enzyme, the activation energy is lowered. So enzymes are not a reactant, nor are they a product. They just lower the activation energy of a reaction, so it helps it occur more efficiently. So exothermic, you end with less energy than you started with, and that difference is lost in the form of heat. Endothermic are endergonic reactions. They're going to end with more energy, right, than they started with. It's going to absorb heat from the surroundings. That's why they feel cold to the touch. It absorbed a lot of heat. And again, if it has an enzyme, then that activation energy, that initial bump, will be lower than if it does not have an enzyme. Another type of reaction that will take place, we have endothermic and we have exothermic or endergonic, exergonic. We also have redox reactions, which stand for reduction, and then OX for oxidation. So oxidation and reduction occur in these reactions. And all they mean is oxidation. A way I used to teach this when I taught chemistry to remember it is oil rig. Oxidation is loss of electrons. Reduction is gain of electrons. So if something loses an electron, that means oxidation occurred. If something gains an electron, it means reduction occurred. So if we look at this molecule A in this diagram, it loses an electron, so it was oxidized. Molecule B gains an electron, so it was reduced. And those terms sound like the opposite of what they should be, right? Reduction, it gained an electron, but it was reduced. Well, its charge was reduced. Its charge became more negative because it gained an electron. So that's why it's called reduction. So with oxidation reduction reactions, again, remember oil rig, and you'll be able to identify what's happening in a reaction. So here we have this electron being lost. So again, that's allowing this molecule to be reduced. So this molecule would be the reducing agent because it's causing reduction to happen. This molecule is gaining an electron, so it's being reduced, but we call it the oxidizing agent because it's allowing oxidation to occur. It's allowing the electron to be lost from molecule X. ATP is super important. We talk about the cell, energy, and even heat, because that's how it can work, is releasing a lot of heat. But ATP, it stands for adenosine triphosphate. The name adenosine, it has an adenine, nitrogen base, has a ribose sugar, and it has 3 phosphates. It looks just like a DNA nucleotide, except it has, or an RNA nucleotide, except it has 3 phosphates instead of 1. So all ATP is it basically is a nucleotide with extra phosphates. And the way it works is these phosphates can be split off, and that releases a ton of heat. And that heat can drive reactions. So you do need to know the structure of ATP as well as its purpose and how it releases energy. Again, it's made of an adenine group, a ribose sugar, 3 phosphates. If that third phosphate is split off, it releases heat, which can drive reactions. I always call it the dollar bill of the cell, because if you want to get anything done in the cell, you have to spend ATP. So it's like spending dollar bills. You want to move sodium across the cell membrane, you've got to spend ATP. You've got to spend those dollars. Once it loses that third phosphate, instead of adenosine triphosphate, it becomes adenosine diphosphate. Because after that point, Well, then it only has two phosphates when that phosphate has been released. So one way they describe it is you'll hear it called a coiled spring, like it's just ready to release that energy when that third phosphate is released. And I just realized the animations are playing on the recording on the computer, but not on the iPad, so maybe you'll see those. So then there's certain things you can add to reactions to make them work better. And one of those is a catalyst. A catalyst is something that lowers the activation energy, so it makes reactions occur faster. So catalysts are not a part of the reaction. They are not a reactant or a product. They just help facilitate the reaction, lower that activation energy, and make them occur much faster. They're not really used up in the reaction. They can wear down, but they're not used up. So they can just be reused over and over and over and over. And we call catalysts in living things, we call those enzymes. So enzymes, they do the same job as catalysts. They're just composed of proteins. So these enzymes, they have a specific shape that determines their specific function. So they follow the same principles as catalysts. The reactants involved in enzyme catalyzed reaction are called substrates, and the point at which the enzyme binds is called the active site. They don't always fit. They don't fit perfectly, so they have what's called an induced fit. And I'll show you a diagram of that here. Just how enzymes and substrates interact with each other. So they call it this lock and key model. If you think about a lock, it has certain pins that need to be compressed to a certain point so that when the key is inserted, it can turn the lock and unlock the door. And that's why the keys have those little notches in them to push the pins up to a certain point. And I used to make keys when I worked at a hardware store. And if you don't get those pretty much exactly right, it's not going to work to unlock the lock. You can have a little variability, but it's got to be pretty darn close to perfect. Enzymes are the same way. So these protein enzymes, they have a very specific shape that correlates to the substrate or the molecule that they want to interact with. So the shape of the enzyme correlates to the shape of the substrate. And the part of the enzyme that interacts with the substrate is called the active site. That's where the activity occurs. So you'll notice the active site shape complements the shape of the substrate. So enzymes can interact with substrates and put them together to build up polymers, or it can take polymers and break them down. But the shape of the enzyme's active site complements the shape of the substrate. And it won't be perfect, so we say it's an induced fit. So when it gets close, it can flex a little bit enough to fit the substrate into the active site, so long as it's close enough. So here are some examples of different enzymes. You'll notice the name of the enzyme oftentimes is very similar to the molecules and the substrates that it interacts with. Protease breaks down proteins. Lipases break down lipids. They even have DNAs, which can break down DNA. Catalase, hydrogen peroxide, lactase breaks down lactose. So again, enzymes are going to lower that activation energy when you have those enzymes. So you'll have some diagrams, and maybe a question I could ask you is, which of these reactions is catalyzed by an enzyme? And you just look, the one with a lower activation energy. What you start with and end with is exactly the same. That doesn't change. It's just how much energy for activation. Now, if conditions are not ideal, then these enzymes don't work very well. So we think about a protein, hemoglobin, for example. It has these 4 tertiary structures put together to form this quaternary structure known as hemoglobin that binds oxygen. This is flowing through your blood right now to move oxygen to your brain, so you can even think about this video that you're watching. If this protein gets too hot, your blood's about 100.4 degrees Fahrenheit. If it gets too hot, maybe you got a fever, you're getting up to 105 degrees, that protein's going to break down in what we call denaturation. So it loses its shape. Well, this shape doesn't bind oxygen. This shape does, but denatured, it loses its function. So our body is trying to maintain these optimal parameters so that our proteins maintain their shape so that they can do their designated designed job. So temperature is super important, pH, salinity, even minerals that are dissolved in the blood can affect how these proteins interact with each other. So your body's doing all of the things it can do to try to keep that temperature and pH at the right ideal conditions, about 7.4 in our blood for pH and about 100.4 degrees Fahrenheit. So again, depending on if these conditions are ideal or not, that's going to affect how these proteins and enzymes function. So if you cool things down, that slows enzyme activity. If you heat them up, it will potentially, it depends on if you're moving out of ideal temperature, well then you're going to decrease its activity to a point until eventually it gets so hot that it's going to bottom out because they denatured. Some enzymes function at different pHs and temperatures based on where they are in your body. For example, pepsin functions at about a pH of 2. That's very acidic. Well, that enzyme lives in your stomach, so it makes sense that it would be designed to function at a pH relative to the pH of the stomach. Amylase works best at a pH of 7, so it's neutral. But that enzyme's in the mouth. So in your mouth, it's mostly going to be a neutral pH, so it's going to function really well at that pH. And then we've got trypsin, which works best at a pH of 9, and this is an enzyme in the small intestine. So from the stomach to the intestine, you have a huge pH change, and that kills lots of little bacteria and infectious particles in our bodies. It helps, nothing can survive, or it's very hard for microorganisms to survive that much of a change in pH. So our bodies have that adaptation to help us destroy these microorganisms. So then we can also inhibit enzyme activity in what we call enzyme inhibition. And there's two different forms of this. You can have competitive inhibition and non-competitive. Competitive inhibition, let's say you've got your enzyme here, and this is its active site where it can interact with a substrate. So this is our substrate. Competitive inhibition is when the competitive inhibitor competes for the active site. It tries to get in the way to block the substrate from interacting with the active site. So it's competing for the active site. Non-competitive inhibitors, they bind to another part of the protein that's not the active site. It's called the allosteric site. The allosteric site is a place where non-competitive inhibitors can bind and change the shape of the active site of the enzyme. So you'll see this non-competitive inhibitor binds to the allosteric site, and then look how small the active site has become. So it's changed shape, and now the substrate can no longer get into the actual active site. It's blocked, but the inhibitor was not competing for the active site, so we call it non-competitive inhibition. And then there's other factors involved. We have cofactors. So these would be non-organic elements such as zinc or copper that could aid in enzyme catalyzed reactions. So that we call them cofactors because they come alongside these enzymes and help them function. Coenzymes, these would be organic molecules such as vitamin B, vitamin C, that also come and aid in these enzyme catalyzed reactions. So if we call it a coenzyme, it's organic. Cofactors are inorganic. And that would be it for today's lecture.